Good morning, everyone. Thank you for coming to this talk. Thank you to those who are presented in the lecture room and to those who joined us online. Today we have Elisabeth Heremans. She is a PhD student in KU Leuven in Belgium, and she is going to present the results of her 3-month-long summer internship in the Audio and Acoustics Research Group, and her talk is titled Shining Light on the Learning Brain, Estimating Mental Workload in a Simulated Flight Task Using Optical f-NIRS Signals. Without further ado, Elisabeth, you have the floor.
Thank you very much, Ivan, for that nice introduction. So I will jump right into my presentation because it was already introduced. This is the contents of the presentation very shortly. So first, let's explain what the words in the title mean, essentially. So we have workload estimation and f-NIRS, which are the two components of my project. Workload estimation means we want to measure the demands imposed by a task on an individual. Why would we want to measure this? Well, for example, in adaptive training, this could be useful because when a person is learning a task, you don't want to give them tasks that are way too easy so that they are not learning anything, but also you don't want to give them tasks that are way too hard so that they disengage from the tasks because they cannot follow or cope with it. So if you give tasks that are just difficult enough, then the learning speed of the individual is optimal. And one potential application for this is pilot training. So pilots are being trained in VR flight simulators that are quite expensive to run. So if we can accelerate the learning process of those pilots in these VR simulators, that would save a lot of money. So this is sort of a scheme of what this project is all about, a schematic. So we see a figure on the left with the pilots in the VR simulator. We capture some signals of this pilot, and what we want to do is estimate the workload based on those signals, so we get some sort of cognitive score. And this cognitive score could then inform an adaptive training system, which will then give tasks to this pilot that are of the optimal difficulty for them to learn at an optimal learning rate. And so in these VR simulators, how can we make the flight task more easy or difficult? For example, we can add mist. In this scenario, for example, the pilot cannot really see the surroundings, or we can add a lot of wind so that the plane is not as stable and they need to do a lot of work to keep it stable. So these are the scenarios that we're talking about, and these are essentially the experiments that we also did for this project. So Professor Dr. Gollard, a previous colleague, actually captured some data with f-NIRS for me to work on in a setup like this. So now f-NIRS. These are the types of signals or brain signals that I will be using in this project. So what is f-NIRS? It essentially measures blood oxygenation in the brain. And why would we want to measure blood oxygenation in the brain? Well, when you are performing a very hard task, you actually send more oxygen to your brain to cope with the demands of this task. So the cerebral blood oxygenation is correlated with neuronal activity. And how do we measure this? Well, f-NIRS works in an optical way. So it consists of sources and detectors. The measurement system has sources and detectors. These sources send two wavelengths of light, and these are absorbed by the oxygenated and deoxygenated hemoglobin, and the rest of the light is scattered, and the detectors pick up some of that scattered light. And by actually measuring the scattered light, we know how much has been absorbed, and by sending two different wavelengths of light and having the oxygenated and the deoxygenated hemoglobin, which have different absorption spectra, we can estimate the concentration of both of those components in the blood. So this is how we measure blood oxygenation in the brain. What do these signals then look like? In an unfiltered manner, this is a very idealized scenario, but this is an f-NIRS signal here on top. which is unfiltered. And there are a couple of components in there, very low-frequent components, low-frequent components. There is the respiration signal that modulates this signal, and then there is a cardiac component, which is a very high-frequent noisy pattern that you see on top of this signal. And so this f-NIRS signal contains both respiration and cardiac information. This is more of a realistic signal. So here you see that we have a number of different channels because we put different optodes and detectors on the head who all measure these f-NIRS signals. And we have an oxygenated hemoglobin signal and a deoxygenated hemoglobin signal. The oxygenated here is red and the deoxygenated is blue. And so for every channel you see we have 2 of these signals. And the heart rate pattern is very clearly present here. So the research goal of this project essentially is to estimate workload based on f-NIRS. How am I going to do this? Well, we have collected this data set with 13 subjects who came in for 5 days, and we recorded them for 22 recordings a day. We collected a bunch of signals, mainly f-NIRS, but also ECG and breathing were collected. Then I'm going to filter these signals, segment them into little pieces, and extract features from them. These features, I'm going to dive into them later in the presentation. And then essentially what we want is to estimate the workload based on these features. And this is performed through regression. So we don't have a classification problem, but a regression problem. We're trying to estimate a score between 0 and 100. And this score is actually the score estimated by the adaptive training system, which tells us how good the subject should be at this task. And so the performance score, of course, is correlated with the difficulty or the workload for the individual. So what we want is have a good correlation between our predicted workload and the ideal score that we get from the ATS, which is our ground truth in this scenario. So our evaluation function is going to be the correlation between those 2. So I already sort of went over this data processing pipeline that I am going to use. And then there are a couple of interesting questions that I want to answer. So first of all, how much do ECG and breathing help when you already have an f-NIRS signal? Is it useful to record ECG, breathing, and f-NIRS? Is it useful to segment the signal into little chopped segments, or can we use full recordings or is it better to use full recordings to predict the workload? Is feature selection useful? And should we filter out physiological noise from the f-NIRS signals? And this physiological noise that I'm talking about, that's the breathing and the heart rate. Or can we use it to actually gain more information, extract more features, and do better predictions? So these are my research questions in short. The data pipeline starts with data selection because we have measured these signals in very realistic scenarios where subjects were in these VR chairs, they were moving a lot. And so there are a lot of movement artifacts which corrupt the signals, and some of these signals are not usable. So first I tried to find some quality metrics that I used per 10-second segments to estimate the data quality and to discard the bad data. And so, as we saw, the heart rate signal should be present in all of these f-NIRS signals. If the contact is good, you should always see the heart rate being present. So therefore, I used the correlation with the ECG and the heart rate power, which is the power in the frequency band of the heart rate. Both of those to sort of see where all the data are and if there's like a secondary Gaussian curve here. These are the data that are actually badly correlated with ECG and that I can basically discard. I did the same for the heart rate. You may wonder why there are some signals with a much higher correlation with heart rate. That is because the heart rate is high frequent noise. So when the signals just have big movements artifacts, this sometimes gives me high correlations with high heart rate powers, so I actually discarded these signals as well. So I only retain the main blobs. So how much data did I then discard out of my total data set? I had more or less 1,051 recordings, and I only retained channels with at least 50% of the segments that were valid. If I have 20 channels and all my channels need to be valid at the same time, I can only retain 20% of the recordings, which is not a lot. So we decided to go with another strategy because of this. If I require only one channel to be valid, so I say I retain the recording if at least one of these 20 channels is valid, then I can retain almost all of my data, but I only have one channel, so I don't have that much information. So what I did was group the channel into groups of frontal, central, and occipital channels, and then a left and a right group. And I require... I think someone should mute their microphone, maybe? Okay, so I require at least one channel of each group to be valid, and that's how I retain 85% of the data and have 5 channels to do the feature extraction on. So then on to pre-processing. So we also need to process these signals in a way that makes it easier to extract the features. For the f-NIRS signals, I use different pre-processing strategies depending on the features that I extracted. But the ECG was filtered between 0.5 and 40 Hertz because that's where the heartbeats are very clearly visible. And the respiration is filtered between 0.1 and 0.5 Hertz because it's a very low frequency signal in that frequency range. And then, of course, I extracted the features. By the way, I didn't say this in the beginning, but if anyone has a question, feel free to interrupt me at any time. So then, after filtering these signals, we go on to feature extraction, which is the interesting part. And this is the main focus of my project, because for f-NIRS, it's really not standardized like in EEG signals, electroencephalography, where there's decades of research on which features to extract. Well, in f-NIRS, it's not as established, and we don't really know which features are useful in the signal. So I had to do a lot of feature design in this project. But first, let's see for ECG and respiration. We extracted 2 basic features, so just the heart rate and heart rate variability, which should be related to stress or workload in an individual. And for respiration, we also looked at respiration rate and respiration variability. So those are the 2 other signals. And now for f-NIRS, here we extracted a whole array of features. So first, I focused on the low frequent part of the signal, which is the main part that everyone else has been focusing on so far in the literature. And I saw this in other papers, basically. So other people already established that these statistical features of the time domain signal of the f-NIRS HBO signal are useful, so we decided to extract them as well. So these are the basic statistical features. Then we can actually also get some information from correlations between the f-NIRS signals on the one hand and these other physiological signals on the other hand. So for example, we know that respiration modulates f-NIRS, that there is some sort of respiration pattern in the f-NIRS, as we saw earlier. So we can look at the correlation between f-NIRS and respiration and the time lag between the 2 signals. Then we have the same idea for ECG, of course, because ECG modulates the f-NIRS signals. We looked at the correlations and the time lags, as well as the heart rate power in f-NIRS. And then lastly, between the oxygenated and the deoxygenated f-NIRS signals, there are also correlations, so we also quantified those and extracted them. And then the next set of features is quite interesting. So this is some sort of synchronous detection feature or set of features where we're using heart rate to segment the HBO signal into little bits and then paste each little heart rate component or each little segment, segmented by heart rate. We paste them on top of each other to get some sort of average f-NIRS signal per heartbeat. Why do we do this? Well, as you can see here, the f-NIRS is sampled at a much lower frequency than the ECG. And so if we want to see, for example, the slope of every single bit here, we cannot extract it very reliably because we only have like 4 or 5 data points per heartbeat. But if we paste each of these heartbeat segments on top of each other, we can get a nice smooth signal and actually look at many features of how an average f-NIRS signal looks per heartbeat. So this is exactly what we did. We segment and we paste all these little segments on top of each other. And we get a nice and smooth, if we average this out, we get a nice and smooth f-NIRS signal, which shows very clearly that there is, for every heartbeat, there's a spike upwards in oxygenation, and then a slower slope downwards when the oxygen is being consumed by the brain, and then it goes up again. And the HBR signal has, so the deoxygenated hemoglobin has the opposite pattern, so it goes down when the heart starts beating, and then it goes up again when the oxygen is being consumed. So this is basically how the brain consumes the energy per heartbeat. So we looked at a bunch of features of these averaged signals. One of them that is interesting is how high are the peaks of every single heartbeat segmented f-NIRS segment. So the peak-to-peak amplitude of the signal is one. Then we can look at the slopes of these signals, and we can model this as a linear function or as an exponential function. The exponential one is more biologically plausible, but it's harder to estimate. So we estimated both. So upward slope, downward slope, exponential or linear. And then we estimated the delay between the HBO and the HBR, because HBO starts increasing before HBR starts decreasing. So we can sort of look at the delay between those two. And then there are a couple more. So the delay between where the HBO signal starts to rise versus where the heartbeat was. This is basically how long does it take for the blood after being pumped to actually reach the brain. And this could be correlated to your cardiovascular system, but it could also be correlated to your workload. We don't really know. So all of these features are hypothesized to be maybe correlated with workload, but we're not entirely sure of this at first. So then there is also the delay between the downslope and the heartbeat, the total rise time, and the rise time divided by the heartbeat length. So all of these are visualized on this little figure here. And then lastly, we have, the standard deviation of these points around every mean signal here. So we have the mean line, and then these points are scattered around it. How big is this variation? Because this is sort of variation between the f-NIRS signals from heartbeat to heartbeat. So the standard deviations as well. Then lastly, we averaged all these features per group of channels, as I already said in the data quality. So we do this because then we can retain more channels than if we use all the channels separately. We can retain more data, because at least one of the channels needs to be good. And then lastly, we do the regression, where we train a regressor to predict the workload score. We use the mean squared error as a loss function and the correlation as an evaluation metric. So let's look at some results here. Actually, the classifier that I did ablation studies with was a deep neural network with 2 layers, 64 neurons per layer. And I used the different modalities first, ablating each one of them to see which ones are the most important. So here you see with all features, we get a correlation score of 0.1. That is, by the way, very low. So we definitely want to bump this up a little bit. If we remove the heartbeat signal, then we actually get a better correlation score. So that means that the heartbeat is not very informative or not very good at predicting workload. If we remove the breathing, on the other hand, the correlation goes down to 0. So the breathing signal is very important. And the f-NIRS signal also is important because the correlation goes down when we remove it. So this is how to read these ablation studies. So breathing and f-NIRS proved important, heartbeat not so much. Then I also did ablations per electrode group. And here we saw that the frontal channels were actually by far the most important. And this is no wonder because your prefrontal cortex is here, so is in the frontal region. So if we measure the frontal region, clearly we pick up on much more about workload than if we measure in the back of the head where your visual cortex is actually. So that's where you're processing visual inputs. So these frontal channels proved sufficient and really the most important for workload estimation. And then my feature groups that I defined, I also tried every single feature group to see which are now the most important features, because these, most of these features are completely unexplored. And so the f-NIRS statistics, the ones that everyone has been using so far, proved really important, so that's a good sign. Then the correlation with the ECG was also really important. And the heart rate power was like medium, but the correlation between HBO and HBR was not interesting to look at, and the correlation with respiration, not interesting. Then when we look at the individual features of these statistics, only the kurtosis and the slope were discarded in the ablation studies, so all the rest are really informative. And when we look at the segmented f-NIRS features, so the ones where we paste all the f-NIRS segments on top of each other based on heart rate segments, we also see that a couple of them got discarded, but most of them are actually very informative. And these are completely new features unexplored in the literature, so we sort of found a bunch of f-NIRS features that everyone has been disregarding so far, but that are really predictive for workload. So that's quite nice. And then when we do these ablations and we only use the most informative features, we shall look at the results in a second. I evaluated this using a couple of different machine learning methods to make sure that everything is sort of stable. My deep neural network is 2 layers with 64 neurons per layer. And I also tried with an LSTM, where I extracted the features in segments. For all the other machine learning models, I extracted features over the whole recording. And I did a hyperparameter optimization for both DNN and LSTM. So here are the final correlation scores using the best f-NIRS features and then trying to fuse with the 2 other modalities. So the top layer is just f-NIRS with the best features. And we see that using the fully connected neural network, we get correlation scores about 30%, 0.32, which is much, much better than the 0.1 that we had earlier. And then by fusing with breathing, we can get to a bit better even, so 0.32, 0.33. So that is quite good. When you compare it to the baseline that we have, where we did the same study with EEG, and with electroencephalography, we got to like 0.13, 0.12. We had a bit better result with LSTM, but this was sort of unexplained, so we're not too sure why that is. But so we can say that f-NIRS is probably more robust as a feature, or as a modality than EEG. And it would be very interesting to see what we can do when we combine f-NIRS with EEG for workload estimation. And then another conclusion here is that early fusion outperforms late fusion for the SVM and the fully connected neural network, but for the linear regressor model, the late fusion is better. So overall, we saw that the fully connected neural network was the best, outperforming all the others. And yeah, that f-NIRS performs quite well at workload estimation. So Conclusions. The focus of this project was to build features, as fNIRS is really an unexplored field. We managed to find some features that are useful for workload estimation and that were completely unexplored in the literature, so we were quite happy about that. A real challenge in this project is that we're trying to build a subject-independent and session-independent objective workload estimator. So when you look at EEG fNIRS literature for these types of tasks, everyone is making it person-specific or session-specific. And that's way, way easier to do because you can calibrate for a certain person, for a certain session. And this is why the correlation scores are relatively low. And then the second interesting or novel feature here is objective workload estimation, because we did not use predefined scores of workload, but we defined the scores based on how well the people are doing at a task. And we can do this in real time, so that's going to be useful for real-time workload estimation. Then what we found is that fNIRS for workload estimation is a good idea. The frontal channels are by far the most informative, and they are enough. So having the frontal channels, we don't need any of the others. We can build much smaller fNIRS devices that just measure like 1 or 2 channels on the forehead. And we also found that simple statistics of fNIRS, the heartbeat segmented fNIRS features and correlation with ECG are good features to use. And then secondly, we found that heart rates is not so very informative. This could actually be because of the way that I'm estimating this. So there might be more robust ways of estimating the heartbeats that I have not explored. So I'm not excluding the possibility that this is because of how we extracted the information and not because of the information in the heart rate. Breathing proved very informative for workload estimation. And ideas to improve this workload estimator in the future to get to more than 0.32 correlation is, first of all, different processing strategies for the ECG to get better heartbeat estimates, potentially, and then combining it with EEG and eye gaze trackers, which are quite easy because they're already integrated in these VR headsets. So we don't even need any extra equipment for that. We can just extract the eye gaze. And eye gaze has been proved to be very informative for workload estimation. So we want to fuse more modalities next time to check how good we can get our system. And then another way to bump up the correlations would, of course, be to make it subject dependent. We made a point of making something subject independent, but definitely having calibrations right before starting the trials, making a very hard task and a very easy task for a person, and then sort of calibrating the scores between those points, of course, helps to make it much, much better. And then, so that's another idea to get better accuracies at the cost of some extra calibration efforts, of course. And then the last step, or the future goal of this whole project, is to integrate this system in pilot training and practice and have a real-time integrated workload estimation system. So that's hopefully also an avenue for future research. Thank you very much for your attention, and I will be happy to take all your questions.
Thank you. Thank you, Elisabeth. Questions, dear colleagues? There is a question online. Feel free to unmute and ask the question. No one? I thought I saw a hand. Okay, before we to start, so usually when we say brain activity, we think electroencephalographic signal. In this work, you introduce a new modality, new ways to register the brain activity, which is indirect. What are the advantages of fNIRS compared to EEG?
Thank you for that question, Ivan. So fNIRS, with respect to EEG, is less prone to movement artifacts. So EEG is an electrical signal, and as soon as the EEG electrodes move a little bit, there are huge artifacts or as soon as the contact is not really good. So it needs to be really in contact with the skin. f-NIRS can still get quite a good accuracy or like performance when it's very close to the skin, and it's less prone to movements. So that's why people have been proposing to use it. It is a less established field than EEG, so definitely there needs to be more work on how to process the signals. But the signals themselves should be more robust.
Thank you. And we have a question from the audience, actually two. Hannes, feel free to unmute and ask.
Hi, sorry, it's a little bit loud here, but I was a little surprised to see that heart rate had no positive impact on correlation. I'm sure reviewers will be surprised to read this. Do you have a theory on why that might be?
So yeah, I looked at my heart rate signals, and I think my hypothesis right now is, so first of all, if we only use ECG, there is a performance, like it's not nothing, so it's adding, so it's definitely doing something for workload estimation, but on top of breathing and f-NIRS, it didn't add any information. That's the conclusion here. So it's not that it doesn't have any information at all. However, I think this may just be the way that I was estimating it, because I used Pan-Tompkins, like a classic algorithm, to estimate the peaks, and then I took the difference between every peak as the heartbeat lengths and estimated the heart rate and heart rate variability from there. So that's a classical way of doing it, but as soon as you miss a peak, your heart rate variability goes up a lot. This way of estimating heart rate and heart rate variability is sort of prone to noise. As soon as there is an artifact, you miss a couple of heartbeats and the heart rate variability goes up. I tried to, when I saw that this was the case, I did some filtering and post-processing to account for that, or like to make it less prone to noise, but still, I think that maybe just looking at the spectrum and checking out the peak that is in the region of the normal heart rate of a person and use the power in that peak, that's probably a more robust way of checking heart rate or heart rate variability. So maybe just a spectral approach would be more robust, and in that case, the results might be very different. So that's my hypothesis here.
Okay,
But also,
got it, thank you.
Heart rate variability is integrated in that noise from which is, when you do the synchronous detection, you compute the standard deviation from the average and from the average line of f-NIRS, and that's also exactly the variability. We have another question online. Feel free to unmute and ask.
Hi. Yeah, I was just asking clarification questions. So you mentioned that you want you pasting all the signal together to smoothing the different frequency. I am a little bit confused on that. Why we not separate different frequency windows and put them different feature together? Why we have to pasting them to make smoothing, and that wouldn't smoothing the how to say the I mean, like that possibly to remove some some of the curves which are not very significant, like those curves. I'm a little bit confused on that approach, why that was more predictable.
I am not sure that I get the question entirely, but I will try to clarify why we did what we did there.
Yeah.
Basically, what we want is to estimate the slopes here. How long does it take for the f-NIRS signal to go up and down with every heartbeat? But we only have like 4 or 5 samples per heartbeat. So you see that this is a very spiky signal with a low sampling frequency in comparison to the heart rate. And so if we cut this into pieces based on every single heartbeat, and then we we have these little segments that we can then sort of shift to be all on the same axis. So I didn't really paste them on top of each other. I would say I shifted them all to be aligned. And then we literally get all these blue dots that you see, which shows a much smoother version of, you can see much more information in there. You can see exactly what the slope is of this f-NIRS signal when the heartbeat happens. Then it starts going up in a very smooth manner because you have all these points and you can sort of average them to get a much better quality signal on a low time scale. So to get a better resolution at this time scale of the heartbeats, that's why we did this shifting. Does that make sense?
Yeah. Thank you so much.
Thank you. Questions? More questions? Questions from the audience? Questions online? Going once? Going twice? If there are no more questions, I would like to thank our speaker again. Thank you, Elisabeth. And thank you for all who attended this final presentation.
Thank you very much.